Hi everyone, my name is Miranda, and today I'm going to talk about animal cloning and a case study, Taiwanese clone pigs. I chose this topic because first, I love the country I grew up in, and second, I read something about this before. Cloning is basically making a genetically identical copy of an organism, and it can both occur in nature and in the lab. There are three kinds of cloning. First, there is gene cloning. Second, there is reproductive cloning. And third, there is therapeutic cloning. But today I'm going to only focus on reproductive cloning, which is producing a genetically identical copy of an animal. So the process of cloning. Let's suppose scientists want to clone a successful sheep A. Scientists take a body cell from sheep A, and then extract its DNA. Then scientists go to find a sheep B, a random female sheep B, and they take an egg cell from it and then remove its nucleus. Then they combine the extracted DNA from sheep A with the empty egg cell of sheep B and uses electric current to fuse these things together. And then the zygote develops into an embryo, and then this embryo is inserted into a foster mother, sheep C. And this foster mother then gives birth to an identical copy of sheep A. But this all really sounds really confusing, so I'm going to use an analogy to explain all of this. Let's suppose sheep A is a successful working kitchen, and sheep B is just a normal or even bad working kitchen. So today we take a chef A and the recipe A from the successful working kitchen A. Then we extract or take away that recipe A. Then we also take chef B and recipe B from working kitchen B. We keep the chef, but we get rid of recipe B. Then we combine recipe A and chef B together. And this is then sent to a culinary school that provides resources and training for chef B. Then the chef B is able to make a working kitchen as successful or even the same as working kitchen A. Then I'm going to talk about Taiwanese clone pigs. The process is basically the same thing, so I'm going to dive in and talk about the somatic cell donor. The somatic cell donor itself was already a recombinant pig. The pig was inserted with the gene that codes for human blood coating factor 9, which is a protein that helps with blood coating. And the pig was also inserted with the gene that codes for lactotransferrin, or LTF, which is a protein that transports iron ions. So these are the advantages of the cloning pigs. First, these clone pigs can produce milk that contains human clotting factor that can use to heal people with hemophilia. Second, the protein lactotransferrin can prevent young pigs from getting diarrhea or getting sick because they use up the free ions that bacteria need to replicate or maintain homeostasis. Pages for further researches on clone pigs that can produce human organs. This is a really big advantage because every day 22 people are dying because they did not have an organ transplant. And second, we can decrease the massive number of people waiting for an organ transplant because now we have pig organs that can replace human organs. There are also disadvantages to this experiment. As you can see in the chart, the number of fetuses that scientists made, and then the offspring alive at the time of publication. There's a big gap between them, and this means that most of the fetuses die. And this makes it unethical because you're killing so many lives. And second, it's unethical to let animals suffer through what they don't have to for no reason. And second, cloning is very expensive. Dolly costed $750,000, maybe because of the successful rate is just so low. And third, this experiment reduces genetic diversity. But I think the pros outweigh the cons because first, scientists might develop ways to increase the success rate and to let animals suffer less. Second, scientists might also develop cheaper ways to do cloning. Third, saving human lives are important. And fourth, I think we have reduced genetic diversity by limiting the amount of modified pigs. And thank you for watching.